Can I start?
Yes, yes.
Okay, perfect. Thanks, Anita, for the nice introduction. So, good morning, everyone. As Anita mentioned that I have a specialization in single molecule biophysics. So I use mainly optical tweezers and magnetic tweezers to study biomolecules and cells. So I will be giving you a brief introduction of how magnetic tweezers actually work. I can't, introduce both optical tweezers and magnetic tweezers in the same lecture. It will take a lot of time. So I am going to focus on magnetic tweezers, its working principle, and I will end up with some examples. So before I actually come to the magnetic tweezers, I thought that I would present few slides on the importance of study of single molecule biophysics, because there are still many people who aren't aware of how single molecule biophysics work and what is their importance. So uh, bulk measurements are very robust in nature and gives you a lot of information, but unfortunately they do an average study of all the measured quantity, and in that lose the distribution of events that are happening, such as some useful kinetics, like for example in molecular motors, if there are pauses, or if there are, what is the processivity of the molecular motor, or what is the speed of the molecular motors, these all things just get lost in those bulk measurement. All right. For example, suppose you are going to a conference and you are delivering a seminar in that conference, and there are some audiences that you take into account. So in this example, the reaction or process is the learning that you are providing to the people. All right. Uh, the sample is whole seminar room. If you are doing the bulk study, you take the whole seminar room as the sample, and the average result is that you assume that everyone learned, right? This is the typical example of the bulk study that people go through. Now, if you start focusing on them one by one, you start to see that someone is busy on phone, someone is sleeping, someone is talking. Uh, there would be various kind of people doing various activities, and in that case, can you say that the average result is everyone learned? No, right? That the average result will change once you start doing the analysis one by one. So this is just a brief, outlook of how single molecule can help you to determine individual pattern inside a study. But coming back to the science of it, uh, in most of the biomolecules, molecules display a fast instantaneous dynamics. They don't have any, average nature associated with all. If you study with single protein molecules or cellular system, you will see that every protein molecule or cell behave in a different way. They might have a similar trend towards achieving an activity, but they will reach to that activity in a different fashion. They behavior appear random and stochastic. As you go smaller and smaller in size of biomolecules, the noise level increases. It is inversely proportional to the population of the sample that you take. So for single molecule, the noise is even stronger. Then fluctuation dominate the behavior of the system, and like for example, in most of the cases, you have molecules suspended inside aqueous medium, or inside our body. They are proteins and DNA are present inside cell, that is again an aqueous medium. So they are under Brownian fluctuation all the time. So there is always a noise associated with them, which again carry a lot of information. So if you do the average of all the behavior, the noise gets also averaged out and become a flat line. That information is lost. Okay. And another thing is that the molecules may be found to coexist in various states. For example, population may be multimodal. So example I am showing you here. Suppose you have two populations in the first sample, which has one distribution is one event is distributed for large number of molecule and shifted towards left, and another is for small number of molecule and it is shifted towards right, and the average is somewhere in the middle. Then you have a single population which is broadly distributed. Its average again is somewhere in the middle. And then again a single population that is narrowly distributed and average is again in somewhere in the middle. Now, if I give you these three samples and tell you to take the average, what you will be seeing just this line in the middle, right? All of them will you will conclude that all the three samples have similar properties. But this is not true. When you do the distribution of events, you will find out that the events are distributed in a different manner than what you were getting in average result. And this is the whole target of single molecule, to identify these kind of distribution and give you a better understanding of how the molecules work. So in this picture, I'm showing you an helicase. I'm sure you would be aware of this typical structure. These two, yellow domains show Recce-like domains, and these in red and brown shows you the auxiliary domains that come out of Recce1. Okay, and there is a DNA that you can see attached to the two Recce-like domains. So this is a extracrystallography structure of a UPF1 structure, and these studies are very useful whenever you are working with any protein molecules in your lab. You want to know the crystal structure so that you can visualize how the protein would work. But as useful as they can be, there is a problem with these kind of studies, is that they are a snapshot of an event that is very dynamic in nature. The proteins are always in some sort of activity, and what you are doing is that you are taking a single snapshot of that protein molecule and presenting it as a study. So you are losing that you are getting the information about the structure, but you are losing the information of the dynamicity of the protein. On the other hand, if you have this gel structure, gel study, just forget about all this, labels. I want you to focus on this gel pattern. If you have a gel study, then you get a lot of information about what the usual trend of the molecule is. But again, you average out the behavior and you lose the kinetic study. So if you see these two type of study, they are very useful, no doubt about that. They give you a wealth of information. They are very robust in nature, but there is something of a great compromise that is happening in both of the case. In one case we are getting a snapshot, in another case we are getting an average value. So there is a need for something in the middle, right? There needs to be something that can fill the gap of this compromise. And for that case, single molecule comes into play. And one of the single molecule technique is magnetic tweezers. So here you can see a typical, representation of magnetic tweezers setup. This is one of the simplest kind of setup that you will see across in biophysical techniques. I understand that most of you are biologists. You don't work with building the setup, but whenever you are working with any setup, for example, a confocal spectrometer or FCS, just try to look at their configuration, how the setup is built. That will fascinate you as well as give you an idea of how these things work. If you compare those setup with these magnetic tweezers setup, you will find that it is one of the simplest one available in the market. So it consists basically of a pair of permanent magnet on an inverted microscope. I will show you the diagram in a bit. So there is an inverted microscope below which you have an objective, and you have a magnetic bead that is attached to a double-stranded DNA molecule, or any other RNA molecule or single-strand DNA molecule, depending on what kind of result experiments you are doing. And as the bead is magnetic in nature, if you bring the magnet close, you increase the magnetic field acting on the bead, and that is how you exert a force on the on the magnetic bead. Now, this magnetic bead is attached to a DNA molecule, which in turn is attached to the surface. So whatever force you are applying to the magnetic bead will be transferred to the DNA molecule. Okay, and this is how we can control the force acting on the DNA molecule inside a magnetic tweezers. This you here again you can replace the DNA with RNA, or you can even replace it with a protein molecule to understand their folding and unfolding pattern. So just a little bit of equation to tell you how these setup works. I promise it is just this one. I'm not going to use anything more. So these magnetic tweezer setup uses super paramagnetic beads. What they are that they are very special magnetic beads that uh, that generate magnetic dipole only when magnet come closer to them. Okay, when there are no when magnets are not closer to them, there are no magnetic dipoles. The magnetic dipoles are there, but they're not aligned properly. But as soon as magnets are approaches these magnetic bead, the magnetic dipoles align with the magnetic field of the magnet, and as the magnet come closer, the dipole moment of the magnetic bead increases sharply. So that is why the force is very strongly dependent on the distance, because as the magnetic dipole increases, the force acting on the DNA also increases. Okay. Now, the property of the magnetic dipole is property of paramagnetic beads are very fascinating in a way that the magnetic dipole aligns themselves with the magnetic field of the magnet. Okay, so the magnetic field and the dipoles are parallel to each other. This is the inherent property. What this means is that whenever you have any rotation that between dipole moment, so mu is the dipole magnetic dipole moment and B is the magnetic field. So whenever and if you do the cross product of this magnetic dipole and magnetic field, what you get is the torque acting on the particle. Okay, this is how the torque is defined. So torque is just the rotational force, if you remember from your high school physics. So torque just means that what is the rotational force acting on a on a particle. So here, if you have mu cross B, then as they are aligned with each other, the cross product will give you 0, because it is a cross product is mu dot B into sine theta if you take the scalar quantity, and theta here is 0, so sine theta will go to 0. So this is how your torque is defined. Now, this gives you an interesting representation that suppose if I rotate the magnet by some degrees, then what will happen is that the magnetic field and the magnet dipole will get misaligned, right? But this is something that paramagnetic bead does not like the equation says. They want to get keep themselves aligned. So what happens is that as you are rotating the magnetic bead also start to rotate with it to keep the alignment parallel to each other. And this gives you a very important feature of magnetic tweezers, that you can do the twisting study of DNA substrate also very nicely with these kind of equipment. This is one of the most unique feature of magnetic tweezers. There are not many setups available in the research field which through which you can do twisting measurements of DNA molecule. So this provides a unique perspective of study. And why do you want to study twisting? Because you have various topoisomerases that are present in our body. For example, gyrases, topo1, things like that, which which remove supercoil. So if you want to study how these DNA supercoils are removed, or how these DNA supercoils are generated, then these twisting experiments comes in very handy. Okay. So the gradient of force determines the direction of force, and this is in the upward direction. Force on DNA increases as magnet move closer to the bead. Okay, as you bring the magnet closer, like I mentioned, the dipole moment increases, and that really increases the force. And as you move away from the magnet, the dipole moment decreases, and you decrease the force. And it can also be used to twist the DNA if it is necessary. Okay. So the forces in magnetic tweezer setup range from a few femtonewton to nearly 100 piconewton. Okay, and this is a huge range. First of all, this is very convenient range because this is the force range in which most of the biomolecules or protein molecules work in. And secondly, this also provides you a big bandwidth. This may seem innocuous if you compare few femtonewton to 100 piconewton. It may seem innocuous, but if you divide 100 piconewton with femtonewton, what do you get? 100,000, right? So it has a value of 100,000 that you can get the force anywhere in between that. So that is a big number. Not many tools are available that can give you that huge spectra of forces that you can apply on a molecule. Okay. I hope this thing is clear. You can stop me anywhere wherever it is not clear for you. Okay. Don't hesitate. All right, so this is the basic theory of magnetic tweezers. I'm skipping the part where you do the calibration and things like that because that becomes a little bit more technical. I want to show you more, applications.
Sorry.
Yeah.
I have a question. So here you've written gradient to force determines the direction. So
Yes.
direction, you mean upwards and downwards movement of the bead or rotation, left-handed rotation, right-handed rotation, something like that?
No, no, no. The force is only upward or downwards.
Okay,
Okay.
okay. So is there a way to control which way your bead will rotate?
Yes. So I will show you in a moment. I will show you. Okay.
Okay, thanks.
So this is how you measure this, the change in the length of the substrate that is attached. And because if you move the magnet up and down, what is you are going to do is that you are also going to increase or decrease the force on the DNA. So when the force on DNA decreases, the DNA tends to coil, and as a result, it comes closer to the surface, right? And when the force on the DNA increases, then the DNA is moved away from the surface. And as a result, the force increases on DNA molecule and the DNA moves away from the surface. So all these things can easily be determined with the help of these diffraction rings. All right. So if you have a microparticle that is present in the focus, you have a very sharp image associated with it. As you move away from focus in minus direction or plus direction, you see that the diffraction ring changes. So basically what we do is that we calibrate the system by keeping a particle stuck to the surface of the flow cell and changing the focus of the microscope objective. Then as you change the focus of the microscope objective, you can see how much change correspond to how many rings, means sorry, not how many rings, what is the diameter of the rings. Okay, you can see that the rings become broader and broader. So you can keep this particle fix, move the stage upward and measure all these variation, and then you have the idea of how much movement corresponds to how much change in the diffraction ring pattern. And then when you have this calculation, you can use it with the DNA suspended in the solution. Okay. Uh, this is the typical arrangement of magnetic tweezer setup. Like I said, it's a very simple setup. You have a inverted microscope on top of which you have a pair of permanent magnet and an LED light that is used to illuminate the setup. Okay. Now, the complication comes a little bit when you have to start doing the manipulation, which you obviously need. So the first thing is that you need to move the magnet up and down to control the force acting on the DNA molecule. All right. So this is achieved by placing the magnets on top of a motorized stage. Usually the requirement is quite strong. You can't, this compromise with the accuracy of the stage because your whole experiment depends on the forces that you are applying on the set on your biomolecule. So you need a very high accuracy motorized stage which you can control precisely to move the magnet up and down. And as a result, you can control the force acting on the superparamagnetic beads. Another thing, like Anita asked, that you can, how do you control this rotation? So this motorized stage is thereby placed on a DC motor. Okay, so this is a rotational motor that again needs to be very precisely controlled. So this motor rotates the, they are, they have some, they are motorized motors, so they can be interfaced with computer, and you can program these motors to provide certain amount of rotation to these magnetic beads. So as you rotate the magnetic beads with the help of these rotary motors, you rotate the DNA molecule along with it. The one condition that you need to keep in mind is that the DNA molecule should be rotationally constrained. That means you should have multiple connection. Okay, it should be a double stranded DNA molecule, and both the double stranded DNA molecule should be attached to the surface of the flow cell. If you only have a single stranded DNA molecule attached to the flow cell surface, then what will happen is that the DNA will not be rotationally constrained. As a result, whenever you will rotate the bead, the DNA will rotate on its free axis, okay, and you won't see any change in the rotational pattern of the DNA. All right. Does that answer the question, Anita?
Yes, sir. Thank you.
Okay. And
Sir,
this
I
is,
have a question.
yeah, please.
Sir, what is the size of these magnets?
Magnets are usually 2 to 3 millimeter one magnet. So you have a pair of magnets. So together there is a millimeter gap in between them. So each magnet is around 2 to 3 millimeter big, and you have a millimeter gap between them. Okay.
And the size of the bead
So the
is
bead
also
size
from that, right?
bead size depends on the experiment that you are doing. So if you have a higher force requirement, then the bead size is usually 2 to 3 micrometer long, 2 to 3 micrometer big. But if you have not so much of a force requirement, like suppose if you are working around 10 to 15 piconewton forces, then 1 micrometer bead will do the job. Okay.
Like, how do we attach this DNA with bead?
Okay. So
DNA is nanometer range, and we are
yes, yes, yes.
attaching it with a millimeter big size
Yes,
bead.
so what usually we do is that we take the excess concentration of magnetic bead to the concentration of DNA molecule, okay, and DNA molecule has this biotin attached to one end and antidig attached to another end, okay. Now we incubate these magnetic beads with DNA molecule, and these magnetic beads has a streptavidin coating. Okay, so the streptavidin coating attracts biotin. So in like 5 minutes of incubation, you can have all the magnetic beads, all the DNA that will be attached to the magnetic beads. Okay, and as you are using excess of magnetic bead, then you make sure that in most of the cases you have single attachment of DNA molecule to the magnetic bead, and the surface is coated with anti-deoxygenane. Okay, so you inject these samples in the flow cell, then these DNA molecules, and you let it incubate for another 5 to 10 minutes. You inject the sample, wait for 5 to 10 minutes, and in those 5 to 10 minutes, your DNA molecules attaches to the surface via dig anti-dig bond. Okay, so obviously you can't observe the attachment in the microscope, because this is a very simple microscope and you're not using any fluorescence dye. So you are correct. It is impossible to see these DNA molecules with your eyes only, but you can observe this attachment, because you have a paramagnetic bead which is under force by a magnet, right? So if you don't have any tether, if you don't have any attachment to the flow cell surface, what will happen is that the magnetic bead will keep on experiencing some force, and as a result it will keep moving upward, right? And then it will finally reach to the top of the flow cell surface and get lost. So if you see that there is some the paramagnetic bead is not going up constantly, that means that it is attached via something to the flow cell surface, right? And that something is the DNA molecule that you provided. Is it clear? Does that answer?
Yes, sir. Yes.
Okay. So let me show you one this video that will answer it better. So you have, for example, DNA hairpin of interest with which you attach a synthetic loop, and here you have a bead binding molecule that is biotin, and this is surface binding molecule that is NTD. People use different chemistry as well. Some people use carboxylic chemistry. Some people use PEG chemistry. So it depends on really what is your expertise and what is your requirement. But the most common one is biotin, streptobidine, and digantidine. Okay, and then you provide some linker to these hairpin substrate. Oh, sorry, and yeah, just a moment. Then you attach the paramagnetic bead on the top, and you have this bottom surface attached to the flow cell. And as you bring the magnet closer, you can increase the force on this DNA hairpin. So DNA hairpin is actually a very interesting substrate, because when it is in folded state, it gives you a double stranded DNA, and when it is unzipped, it gives you a single stranded DNA. So you can very easily play between double stranded and single stranded DNA molecule. Okay, and unzipping of these hairpin substrate can be done either it is a double stranded DNA molecule, so you can use a helicase to unzip it, or you can use a high amount of force, something around greater than 15 piconewton, can unzip a hairpin molecule from double stranded to single stranded. So you can do this. Here I'm showing you an example which you can achieve with the help of this force being applied to this magnetic beads. Okay. Now, the benefit of using magnetic tweezers is that you can do this for hundreds of beads at the same time. So, for example, you design your flow cell, you inject the sample in your flow cell, and as the surface is coated with anti-decoxygenase, many DNA beads will attach randomly to the surface. Okay, and you can apply the force on all the DNA beads in the field of view of your microscope at once, and observe the unzipping and zipping pattern of your hairpin substrate at one go. Of course, this is a simple experiment that I'm showing you here, just application of force to do this unzipping and zipping. But likewise, you can do protein DNA interaction of these hundreds of molecules at the same time. Okay, and do the kinetics of these individual molecule one by one. So this is another benefit, because it is a single molecule benefit, but it is relatively high throughput method. All right. So coming to the application of these studies, here I am showing you the helicase activity of UPF1 helicase, the one which I showed you the crystal structure. And I will explain to you what is happening here one by one. But remember in the video, as I mentioned, that you can unzip this DNA hairpin by two ways, either by applying the force or by using the helicase, right? So what is happening here is that this yellow oval shape cartoon represent the UPF1 helicase in presence of ATP molecule, and this is the DNA hairpin substrate, and this DNA hairpin substrate is under constant force. Okay, so you keep the magnet at a fixed location, that gives you constant force being applied to the substrate. Now, as the helicase attaches to the hairpin substrate, it starts to chew on the double stranded region, right? It starts to unzip the double stranded region. So it starts to move forward, unzipping the double stranded region. And as it unzips it, what is going to happen is you will start to observe a change in the length of DNA substrate. And this change of length in DNA substrate can be accurately measured with the help of the images diffraction ring that I showed you 2 slides ago. Okay, so as it keep on moving, it reaches the loop. It reaches the loop of the hairpin substrate, and it does not fall off here. It keeps on walking on the single stranded region. So this portion is called the unwinding of DNA molecule, but it keeps on walking on single stranded region, and it starts to do a single stranded translocation activity until the DNA hairpin is completely zipped. Okay, and it reaches to the end of the substrate and eventually falls off. So this behavior can be seen here. Okay, so when it starts to unwind, what you this is the position in the graph which is being shown in this location. So this picture shows you the event happening in micrometers. So most of the time, the results that you get are in micrometer, but you can easily convert it in base pair with the help of the calibration that you do with your microscope. Okay, the conversion is very straightforward. But just for the sake of simplicity, just assume 1 micrometer represent the unwinding of one base pair. That it's roughly this equation. One base pair is 1 micrometer. Sorry, 1 nanometer. So this is a 1.2 kb hairpin substrate. So this comes around 1 micrometer of unwinding. So as you can see that at this location, helicase comes and binds, and as it starts to unwind, you see an increase in the length of the DNA molecule, like you see here. Okay, and when it reaches the loop, it reaches the peak of the unwinding. So here you have a single stranded DNA present, and then it starts to translocate on this substrate. All right, on single stranded DNA molecule. So this simple experiment gives you a lot of information, right? First of all, it gives you the speed at which the helicase works. So for example, during unwinding and during translocation as well. And then it tells you that the helicase has a very strong grip on this substrate because it can keep on unwinding. It reaches the top. It goes on in a single stranded DNA molecule, but it does not fall off. That means it has a huge processivity. So you do this for hundreds of DNA substrate because this can be one-off event, right? You can have one particular helicase that gives you this processivity, but you do it for hundreds of different substrate, and you will find that this is a common trend that this helicase have very high processivity associated with it. Okay, so with this simple experiment, sorry, with this simple experiment, you get wealth of information about the property of the helicase. Now, this gives you a simple understanding of how the helicases can be studied, how the processive helicases in a single molecule setup can be studied with the help of magnetic tweezers. But you want to do more, right? You want to understand how the functioning happen, what makes these helicase processive, what makes another helicase less processive, and what the mutations can lead to. Like for example, this is a very important helicase because it helps in the regulation of nonsense mediated decay. So it helps in the whenever you have pre-messenger RNA that is not properly coded, then it helps in the destruction of those RNA molecules, so that immature protein is not formed. So this is a very important helicase. From the same family, so as you already have studied helicases, you must be knowing that there are 6 superfamilies, and then in those superfamily you have many subfamilies, right? So in one of the subfamily called UPF1 like helicases, there are like 9 different helicases that are present. And in that you have two similar helicase like UPF1 and IGH-MBP2 that has very conserved structure. They have similar Recce-like domain, so Recce1, Recce2. The only difference, so this is called the helicase domain. Recce1 and Recce2 is called the helicase domain. These things that are protruding from helicase domain are called the auxiliary domain. So if you concentrate only on the helicase domain, you will see that both of them have very conserved structure. Only difference is that in the auxiliary domain, you have 1B and 1C that is protruding out from Recce1. This UPF1 has different loop that is present at 1C. So there is a loop structure that connects this Recce1 with 1C, and this loop has more binding site to DNA as compared to the loop of IGHMBP2. Okay, so the binding sites in the loop region is different. That's the only difference between these two helicases structured. So if you study these two helicases in magnetic tweezer, you see a very strange behavior. One of the helicase, like this UPF1 helicase on the left, both are helicase domains. So without these auxiliary domains, so this the UPF1 helicase shows you a high processivity. It does not fall off. But on the other hand, IGHMBP2 is dead. Okay, it does not show any opening event at all. There is no unwinding event that takes place. So that led us to question what is going on. Why one of the helicase with similar structure acts like a super helicase and another one acts like a dead helicase. So like I mentioned that they have a loop structure, and when we explored that loop structure, we found that for UPF1 there are 4 amino acids that is in touch with DNA molecule, but in the case of IGHMBP2 there is only one amino acid that is in touch with DNA molecule. So obviously, if you have more attachment points with DNA molecule, that gives you better, binding affinity of one helicase to the DNA molecule. But if you don't have this attachment, if you have a single attachment point, the affinity is weaker. So to understand this binding affinity, you can do a simple assay. Uh, for example, here I'm showing you, uh, force cyclic assay. Just bear with me. I will explain each and everything in this diagram. So on top graph, what you see here is the force acting on the DNA molecule. So in this brown curve that you see here, and at the bottom you see is the extension of the DNA molecule happening. So there is there is a cyclic nature. You increase the force so that the DNA hairpin unzips. Then you decrease the force to a certain level. Okay, so for example, in this case, it was 7 piconewton, and you wait for at that force for some period of time, and then you go to 0 piconewton. Okay, and you repeat this cycle again and again. So what is going to happen is that when you increase the force above 15 piconewton, like I mentioned that at 15 piconewton this goes to double stranded DNA. Sorry, double stranded goes to single stranded DNA in case of hairpin. Okay, always remember, in case of hairpin this is the case. Now what is going to happen is that when you unzip the double stranded DNA molecule, and if you don't have any ATP, just the UPF1 present, and when you reduce the force to 7 piconewton, what is going to happen if the UPF1 binds to the to the single stranded region? Because it has a strong grip, if you reduce the force, the hairpin will not close. Okay, as you can see here, in the first cycle the DNA is unzipped, and when you reduce the force, the DNA zips quickly because there was no UPF1 present. But as UPF1 comes into the DNA substrate, it remains bound even if when you're reducing the force to 0 piconewton. See what you are doing when you're reducing the force to 0 piconewton, you're forcing the protein out, because this fork, the DNA closing fork, is going to push the protein out of its system, right? There is always a force acting on the protein molecule, but the binding is so strong that even at 0 piconewton, the bead is the hairpin is unable to kick the protein out. And as you don't have any ATP molecule present in the substrate, all you can see is that the protein stays at the same location for a long period of time. So this measurement was done for like 3000 seconds, and we did not observe this protein molecule to fall off. On the other hand, if you see IGHMBP2, you can see that for every cycle, IGHMBP2 binds to the hairpin substrate and keeps falling off. Sometimes it falls off in some in long period of time. Sometimes it falls quickly, like for example here. Sometimes it does not bind at all. So for similar concentration of both the helicases, we see two different binding affinity. Okay, and this binding time you can calculate easily. This is a two-stage process, so it follows an exponential decay, and you can fit the exponential decay to get the average binding time of 22 seconds for this IGHMBP2. Okay, so this grip was something that we thought that is responsible for the high processivity of UPF1 and low processivity of IGH-MBP2. So to do an experiment, what we did is that we exchanged this, AKS mutant, which HPA mutant in UPF1, and we did a triple mutation, and surely we saw a huge reduction in the processivity. So earlier, UPF1 has 10,000, more than 10,000 base pair of processivity. But in this case, once you do the mutation, the processivity reduces to 37 base pair. Okay, so this loop was definitely one of the reason that was affecting the processivity of this two helicases. And as you can see, if you do the same experiment of cyclic assay, you will see that the grip also becomes loose for this helicase. You can see that the DNA, the helicase is still having binding strongly to the substrate, but it is being pushed out. Whenever we are doing this, closing of the hairpin loop, the loop pushes the protein molecule out of it, out further and further. So this is almost, similar to you go on the top of a marble pole, and you can wrap your hand and leg around the marble pole. So to hold it tightly, but because the nature is because the friction is less, it becomes slippery in nature, and you slowly keep on going down because of the gravity. So likewise, the grip is still strong, but it is slippery in nature. Okay. So this was one study. We also did a reverse study in which we met the IGH-MBP2, which exchanged this mutant, these three amino acid with these three amino acid in IGH-MBP2, and we found that the dead helicase became a super helicase. Okay, so these studies we were able to show that how the processivity and the grip of one helicase is affected by the binding affinity. So this tells you a lot of how interdisciplinary studies, for example, keep taking the structure from crystallography and application in single molecule gives you the complete picture of what is going on with the helicases. Okay. In another example, I just want to show you how you can also study higher order structure, secondary structure in DNA substrate. For example, here you have uh, G4 forming motif. I hope you all are aware of what G quadruplexes are. It is just a higher order structure that is formed between guanine rich DNA molecule. So if you have a repeat of guanine molecule, it forms non-Hookean, non-Watson-Crick base pair called Hookstein base pair, and that gives you a very stable G quadruplex structure. Stability depends on various factors like temperature, pH, and the cation that is present in the buffer. For example, it is more stable in presence of potassium than in presence of sodium. So this is also a very important structure that people are studying a lot these days, because G4 are not easily resolved with the help of helicases. Most of the helicases are not able to resolve G4. So suppose if your gene has this secondary structure present, and there is a replication cycle that is going on, and in that replication cycle the helicase encounters this G quadruplex substrate, what is going to happen? This will create a kinetic trap, because the helicase will not be able to resolve it, and it won't be able to move forward. So your cell will not be replicated. Because your DNA is not replicated, your cell will not replicated, and eventually it will die off. And this process is being utilized in drug delivery mechanism for treatment of for say cancer molecules. So that's why these G4 structures are becoming more and more a topic of study in many labs. Of course, there are a few helicases that can do this unwinding of G4 substrate. That's why the replication can occur. But the whole idea is how we can stabilize it more and more to get to prevent the helicases from unwinding it. So here you can see the formation of G4 substrate in a hairpin molecule. You have a G4 binder forming motif. And when you go to the single stranded region, and when you reduce the force, it is possible that G4 motif gets into a stable G quadruplex structure. Okay, so sometimes it can be possible that if you reduce the force even further, then there is a loop formation where the hairpin substrate forms the double stranded region over the G quadruplex structure, but this is very rare actually. This does not happen very often. So like in the case of UPF1, you know, like in the case of this UPF1, this substrate will also provide a roadblock. So the hairpin will not close completely. It will stay somewhere in the middle. The hairpin will is closed partially, and this partial closure will be seen, okay, in your magnetic tweezer setup. So what I'm showing you here is again the unwinding and rezipping of hairpin substrate in presence of UPF1 and ATP. And like you can see in this example, the hairpin. So this is a hairpin with a G4 forming motif. So as you can see, the hairpin is able to sorry, the UPF1 is able to unwind the hairpin substrate, and as it starts to go on a single strand region, it encounters, because once the hairpin is completely single stranded in nature, it can form G4. Okay, and as it starts to go on single stranded region, it encounters the G4 substrate, and as you can see that it tries to unwind it, but it remains there for a long period of time, and during that time another UPF1 helicase can come, and because you still have a partial hairpin structure, right? So another UPFN helicase can come here and start to unwind this partial hairpin structure. So this unwinds the partial hairpin structure, and again, it is not able to unwind G4 region. So this example I am showing for UPF1 because, like I showed you before, UPF1 has very strong binding affinity with DNA molecule, but even high binding affinity means nothing when it encounters uh, some some secondary structure. So it has a specific job of unwinding double stranded RNA and double stranded DNA molecule, but it is not meant for unwinding this secondary structure, and that you can see in this in this experiment. On the other hand, yeah, so this is how it looks like. You have G4 present, and this G4 is unable to be resolved with the help of UPF1 helicase. On the other hand, you have another helicase called PIF1, and this has been known historically to resolve G4 structure. And you can see this helicase has a very dynamic behavior. It unwinds, it slips back, it unwinds, it slips back, and it is not as smooth unwinding as UPF1, as you can observe here. And it reaches a place where the G4 formation has taken place, like you can see here somewhere. And depending on the stability of the G4 molecule, it takes certain amount of time. For example, here it is taking something close to 20 seconds. So if you wait for this helicase, it takes around 20 seconds to resolve this G4 structure, and then it starts to unwind the double stranded region even further. Okay, and then it falls back, it starts to unwind again, and so on and so forth. So from this in these traces, you can calculate the average time. So you can do this G4 formation and resolving of G4 for multiple cycles, and with these experiments you can calculate the average time that PIF1 takes to resolve G4 substrate in a DNA substrate. So you get valuable information of the time required for resolving these G4 substrate. What you can do in these experiments further that you can use an aptamer or a ligand that stabilizes G4. Okay, because the whole idea is to use these substrate as a drug delivery system to target cancer cells. So you want to keep these G4 substrate very stable. So you can increase the stability with some ligand or some aptamer, and then again introduce PIF1 in the system and see whether the average time that it was taking to resolve G4 before the stabilizing agent increases or not. And these behavior can give you yield a very accurate time representation of how the PIF1 behaves in presence of certain ligands. Okay. And one final example that I want to show you is this the use of SGS1. That this is a helicase from Recu family, and they are again very important helicase because they are known for DNA strand repair with the help of homologous recombination. And in this example, instead of using a DNA hairpin, what they used was a nicked DNA. So in double-stranded DNA has a nick, and a loading region of 40 nucleotide long, this single-stranded region. So you must be aware that most of the helicases need a single-stranded loading region. So that's why for hairpin, you have a natural single-stranded loading region with the linker. And in the case of this nicked DNA molecule, they need to provide a loading region with the help of single-stranded DNA. So here the SGS1 just comes and binds, okay? And you can observe that there are various regions in this this helicase unwinding. It unwinds and falls off, and it also shows you bursts. So for example, here you can see that it goes on very quickly. There is a pause that is taking place, and then it starts to go in the burst region. So all these pauses and bursts that you can quantify very accurately with the help of these kind of studies, which unfortunately gets lost when you study their behavior in presence of bulk. In, like for example, you see the unwinding in gel, you will not get any information like this. Now, if you attach RPA with SGS1, what turns into a very interesting pattern, like here, you can see that the helicase unwinds and it slips off. Okay, it does not translocate like UPF1. It slips off. Every time you see it slips off. But when you add RPA, it unwinds, and then it goes under single strand translocation. Okay, and then you also observe is that there is a strand switch. So it starts to unwind. It unwinds. It starts to translocate on single stranded region, and there is a strand switch that is taking place in this UPF1. So it goes from 5' end to 3' end, and then again it starts to unwind the double stranded region and then go into single strand translocation activity. And you can observe the speed and this property of the helicase also changes to some extent. Which is expected because RPA is a single stranded binding protein. So definitely it will alter the behavior of the helicase. But what is important is the strand switching activity that you observe, which was absence in the case of just SGS1. And the authors argued that because this is a DNA repair protein and it works by homologous recombination, the strand switching can be an important aspect of that homologous recombination that is responsible for this kind of behavior. So with this example, I think I am done with the lecture on magnetic tweezers. Let me know if you have any question from any of these experiments or regarding the working of the magnetic tweezers.
Thank you so much, sir, for this wonderful introduction to magnetic tweezers. Students, questions from you.
Good morning, sir.
Good morning.
So I have just a confusion about this burst, like as we just explained in this burst graph, in this graph, like this burst part. Can you please explain it once more?
This one you mean?
Yes, sir.
Okay. So what is happening here is that this helicase is unwinding. Okay. So you in this case the mechanism is a little bit different. In hairpin it is quite easy that you go from a double stranded to single stranded and you see the increase in length. Here also you go from double stranded to single stranded because when it starts to unwind, your double stranded DNA goes to single stranded region. But the length change is means the parameter is different. But if you observe any double stranded and single stranded DNA molecule, you will see that the force acting on this single stranded The extension of single-stranded DNA molecule at the given force is higher than the extension of double-stranded DNA molecule. So the extension changes as the helicase is unwinding, right? And that extension change is observed here. This is fine. I hope you understand that. Now it reaches a certain position and then it starts to go on a single-stranded region. So it starts to do single-strand translocase activity. Okay. And as it was going to single stranded region, what happens is that at that point, the helicase switches the strand. So suppose it was going on this strand, it goes and switches to this second strand. Okay. So this strand switching leads to, so suppose it was going in the forward direction, when what will happen when the strand will switch, it will start to go in the backward direction. All right. So again, when the strand switches, it starts to unwind again, and this and reaches the top, and then starts to go to the single strand translocation activity. So this strand switching activity you can observe actually at many places here that they have shown that it goes, it starts to go back, and then again start to unwind, comes start to translocate, and again starts to unwind. So this simply means that it goes from one of the DNA strand to another DNA strand. Okay, and as a result, its direction changes. Is it clear?
Yes, sir. Yes, sir, it's clear. Thank you.
Anything else?
No, sir.
Any other questions? No. No questions anymore?
Sir, I have a question.
Okay.
Sir, in the beginning, you show us a video. In that video, like how is the magnetic field is maintained like uniformly? Like there were like thousands of beads. So like how for that, like how this magnetic field is applied on all of them equally?
Okay,
Like
okay.
if there is one this magnet, then that will not create the field which will be uniform everywhere.
Yes. That's a good question actually, and valid concern. So see, like I mentioned, that the magnet is something around 2 to 3 millimeter, right? So together they have a length of something close to 5 to 6 millimeter. And if you include this slight gap in between, then it is something around 6 to 7 millimeter. What is the usual field of view of your microscope? It is something close to 300 to 400 micrometer for a 100x objective. So the objective used here is usually 100x to get the better images from the diffraction ring. So for that, 100 to 300 micrometer range of field of view is very small compared to the size of the magnet. Okay, it is almost millions of magnitude smaller. So this is why the field is for all practical purposes you can take the field magnetic field over that field of view as uniform. All right, and that's why we consider the forces acting on each particle in the field of view is constant. Do you get the point?
Can you please explain the last point again? My internet was I have some internet issues and I
Yeah,
couldn't.
sure. So what I was telling is that your field of view is 100 micrometer, and you have a you have a magnet that is something 3 to 4 millimeter big. Okay, so if you consider the field of view with respect to the magnitude of the magnets, this tells you the field of view is very small, and as a result, for all practical purposes, you can consider the field to be uniform for the given given field of view of the microscope. So that's why we consider it as a uniform magnetic field in the field of view in which we are taking the measurements.
But when there are thousands of such beads, then how can that be? Like
Yes,
for
so
one bead, I am understanding.
yeah, but the point is all the thousand. So see, we have this. Okay, this is what you are mentioning, right? So
Yes, sir, exactly.
we have the thousands of beads present on the flow cell surface, but you are not measuring thousands of beads at the same time. You are measuring only the beads that is present in the field of view. You don't care about what is happening to the beads that are not present in the field of view, whether they are unzipping or not zipping. So you are only focused. So even if you focused only on field of view, you can study hundreds of molecule at the same time, because the bead diameter is only 0.5 micrometer. So sorry, 1 micrometer. So bead is very small. So even in 100 micrometer by 100 micrometer field of view, you can get something close to 100 different polystyrene beads that are present. Okay.
Yes, sir. Yes, sir. Understood. Thank
All
you.
right. Anything else?
I just have one question, Saurabh.
Yeah.
So, the helicases that we study in single molecule, we know that their processivity might actually increase if we consider the additional other proteins that they interact with.
Mm-hmm.
Like it happens inside cells. So,
Yeah.
but given the nature of this setup, is there a limitation towards using say multiple proteins? Like in the last slide, you showed that there are other protein complexes that you added to, but can you actually study large protein complexes using such setup?
Yes, sure. And this actually becomes very beneficial. Suppose you have more than one protein complexes that is acting on it, acting on your DNA molecule. So that is also very easy to study, and that gives you much more information, because suppose you have say in case of helicases itself, you have one helicase, but it does not work with the help without the cofactor proteins. For example, in case of this. In DEAH family of helicases, you have PRP43 that works in the spliceosome and in ribosome biogenesis. And these helicases don't work alone. They always need some cofactor to work with. So you can always, work alone with the helicase, see whether they have any activity or not, and you can introduce each cofactor one by one, and then you can see how these cofactors are affecting the property of the helicase if they are being introduced one by one, and that gives you even more information about the cooperativity of all these protein molecules in presence of DNA substrate.
Okay, and one last question. So, you have showed us a nicked DNA strand that could be loaded with a helicase, and then the activity could be studied. You've
Yeah.
also showed a hairpin-like DNA structure that could be used to load a helicase. Are there any other structures, say a Halliday junction kind of thing, which is used in recombination? Any other structures that can also be used similarly to study under a magnetic tweezer?
Yes, so people have used holiday junction as well, and this you can use RNA molecule or with this setup as well, double stranded RNA or single stranded RNA molecule. Only thing is that as you go with RNA, the complexity increases, because you are using a very uh, a flow. You are using a lot of things in it. You are using syringe, you are using pump, you are using flow cells. So there is a lot of chances of getting slight contamination. So you just need to be extremely careful in using the RNA. You can also use a single stranded DNA molecule, or you can use double stranded DNA molecules. For example, if you're using studying topoisomerases, you need double stranded DNA molecule to study its behavior. So and you can use protein substrate. So it really depends on it is really limited with the imagination, means how much you can imagine your substrate to your substrate to be like. If you can have the imagination of making a substrate, it is possible to do that.
Okay, and one last but very naive question. You have clearly showed how you track the forces and the distance that this DNA has moved up or down. Look by just looking at the diffraction ring sizes
Mhm.
of the bead. So as it moves off focus up or down, you see changes in the diffraction rings. You measure that, you know the size. But what about if the bead is rotating, is that also measured somewhere?
What do you mean when you rotate the bead?
So, because the bead is fixed in space with the help of these magnets, and you're able to look at the translocation of the bead either up or down.
Right.
But you also mentioned that it is because it's paramagnetic, you might also be able to rotate the bead.
Yes. So again, this is done with the help of change in length. Okay. So like I mentioned, it needs to be torsionally constrained. So you need both the strands of DNA molecule attached to the surface. And if that takes place, then when you're rotating the DNA molecule, maybe I can show you one slide. Just give me, that will be better. Just give me a minute.
Sure, but very sorry for taking so much time.
No, it's okay. Sorry, I need to find, yeah, this one will do. So yeah, so this is how you measure it actually. So if you rotate the DNA molecule, depending on the force that is acting, so less than, if the force is less than 0.5 piconewton, what you are going to see is that as you rotate the DNA molecule, you're going to create platinums, like the braided hair that you see. So as you create the platinum, the length of the DNA molecule decreases, right? The DNA was elongated; it has particular length. When the platinums are created, the length reduces, and then again this is reflected in this diffraction rings.
Diffraction.
Okay? Okay. So you can measure, like for example here. So this is a typical rotational measurement for length less than, for force less than 0.5 piconewton. Remember, in case of rotation, the force becomes very important. So maybe we can discuss about it later on, because it's again, it will take a lot of time to explain this thing, but you can measure this length change very accurately with the help of diffraction rings. When you are below 0.5 piconewton, you can see that you can do negative as well as positive supercoil, generating positive and negative platinums in your double stranded DNA molecule. So this is the extension that is being shown, right?
All right. Thank you, Saurabh. Thank you for patiently answering all our questions, and thank you for this nice introduction to magnetic tweezers. I'm sure our students have learned a lot today, especially given that you've explained it in such simple terms. So thank you, students. Let's all thank Professor Saurabh Raj for taking time off his busy schedule to give us this lecture. Thank you, Saurabh.
Thank you.
Thank you, sir. It was really a very nice and simple presentation, and we were enjoying it. Thank you.
Thank you. Thank you.
Okay.
Okay. Okay. Bye-bye.
Bye-bye. Bye. Thank you, students, for joining in. We'll have our next class on Friday. Thank you. Bye-bye.